Hello, my name is Nadav Ahituv, and I'm a professor in the Department of Bioengineering and Therapeutic Sciences, and also the director of the Institute for Human Genetics at UCSF, University of California, San Francisco. And I'll tell you today about what we're doing in terms of developing a potential therapy for Cri du Chat. And so, in order to explain this, I'll first talk about regulatory elements, which my lab specializes in. So, you probably have all heard of genes, and genes could be envisioned as a light bulb. And if that light bulb, in order to turn it off, it has a regulatory element right next to it called a promoter that basically turns it on, and simple light switch on off. And in addition to that, in the DNA, we have what's called enhancers, which are sort of the dimmer switches that tell that promoter to turn on at specific time points and specific locations, and combined give us this complex expression pattern. And so why am I telling you all that? Well, what we do in the lab is take advantage of these switches to basically change the expression of the gene, for example, increase the levels of the gene. And through that, we could use it for therapy purposes, as we hope to do here for Cri du Chat. So just to show you an example of how this works, we first did this on diseases called haploinsufficient diseases. It's a long word, but haplo basically means half and insufficient is not enough. And basically what we have is two copies of a gene in our genome, one from our mom and one from our dad, and that leads to normal gene levels. Every so often, we could have a loss of function mutation where one copy is not enough, and that leads to 50% of a gene being made. And surprisingly, this is fine for many genes, but some genes, and we've looked through the literature, there's 660 genes where having half is like running on empty, and it leads to human disease. And so I'm assuming you've all heard about CRISPR. So CRISPR basically is a tool that can come to the DNA and make specific changes or cut out specific regions and change them. But what we've been using is a different kind of CRISPR where there's a mutation to the scissors, so they cannot cut the DNA, but we can use them as a delivery truck to bring something to a regulatory element, for example, here to a promoter. And that something is an activator that will force it to make more RNA and more protein, and that's being called CRISPR-A for CRISPR activation. So going back to the model here, what we could do is there's still that existing copy right here that's functional, so we can target a regulatory element of it and force it to make more RNA and more protein, and by that rescue these diseases that have a low dosage effect. And we've shown this now in the lab for a few diseases. This is an example for obesity, where you can see this mouse has one copy on the gene here on the left. The mouse in the middle is the same genotypes, has just one copy of this gene, but because of the CRISPR-A therapeutic approach, you can see that it looks like a normal mouse on the right in terms of its body weight and is not obese. So what are we doing in terms of Cri du Chat? Well, Cri du Chat is basically a deletion of the short arm of chromosome 5, and in that deletion, there's over 90 genes that are missing. Now, not all these genes, if you have one copy of them, this will be a problem. And so what we started doing is working on one gene, CTNND2, which we think is going to be the main gene that's important for neurological phenotypes in Cri du Chat, and to try to upregulate it in terms of this approach to see if that could be helpful. To do this, we're optimizing the conditions for this, and then we hope to test in stem cells from patients to see if we can upregulate this gene. And we've also initiated collaborations with the COSIC lab at the University of California, Santa Barbara, that have organoids with a 5p deletion to see if we can change, and organoids are basically sort of small brains grown in a dish, to see if we can change any of the phenotypes in these organoids. And all this work is being done by Deanne Leboy-Citron, an outstanding graduate student in my lab, who will talk about her work on this project. And I'll pass it on to Deanne. Thanks.
Hi, my name is Diane, and I'm a graduate student in the lab of Dr. Nadav Ahituv, and I have the pleasure today of talking about the work we have been doing in the lab to develop a CRISPR-A therapy for Cru-du-Chat syndrome. I would like to begin by thanking the Cru-du-Chat Research Foundation for their research funding, and also thanking Dr. Nadav Ahituv, my research mentor, for allowing me to be part of this project and for his commitment to this research project. There are three main things I want to share with you today. First, what we have been doing to optimize the CRISPR-A therapy for CTNND2. Second, our optimization for the delivery of the CRISPR-A therapy through AAV into neurons. And third, what we have been thinking and the sign of experiments that we're going to be doing to test the efficiency of this CRISPR-A therapy in patient-derived cell lines. So I first want to share about the work that we've been actively doing to develop the CRISPR-A therapy for CTNND2. So even though there's a lot of genes in the 5p minus deletion, why specifically are we targeting CTNND2? CTNND2 is a haplosufficient gene. As Dr. Nadav Ahituv mentioned, a haploinsufficient gene means that one copy of the gene is not enough to do its work. Second, we also want to target genes in the 5p minus deletion that has a clinical relevance for Cru-du-Chat syndrome. CTNND2 is one of those genes which is attributed to lead to neuronal phenotypes observed in Cru-du-Chat syndrome. And third, something that is important is knowing a little bit about what a gene does. And I want to share a little bit about what specifically CTNND2 is doing. So CTNND2 is primarily expressed in the brain, and specifically in the cells that make up the brain that, as you might know, are known as neurons. And neurons are really interesting because they have a unique shape in which the nucleus and other things that make up the cells are concentrated in a region, and they have this long projections that allow them to make connections with other cells. In these connections, CTNND2 actually lies in, and it aids in the process of cell adhesion. And also, through the cell adhesion role, CTNND2 is important for allowing the cells to migrate during brain development. Another role that CTNND2 has is as a regulator of genes where it allows them to turn on and off. So in summary, CTNND2 is a gene that lies in the 5p minus deletion. It is haploinsufficient. It is hypothesized to lead to the neuronal phenotypes observed in Cru-du-Chat. And we know a little bit about what CTNND2 is doing in the brain, which makes it an ideal target for the therapy that has been developed in the Ahituv Lab. So the main thing that I've been doing over the last few months is designing and testing guide RNAs to target CTNND2. And you might be wondering, what are guide RNAs? So as Dr. Nadav Ahituv mentioned, our approach has two main aspects of it. It has an activator and a delivery truck. So I like to think of guide RNAs as a map that tells the delivery truck where to go and therefore takes the activator to a gene to turn it on. We use a publicly available data set developed in the Broad Institute known as CRISPRa to design the guide RNAs. Here I have a representation of the CTNND2 gene. This is how we represent genes mostly, and what you need to know is that this gene, denoted by these arrows, goes from right to left. Something really unique about some genes is that they come in like different forms, and CTNND2 is one of these genes. Data from one of our lab members showed that CTNND2, the long form, is one of the most important in the brain. And therefore, we design our guide RNAs to that long form of the CTNND2 gene. So basically, what the guide RNAs that we design do is like they will act as a map, and they will tell the delivery truck, go to this gene, and we'll get the activator into that area and turn up the expression of that gene. In the lab, I tested the regulation of 15 guide RNAs that are denoted here, with the control with no guide RNA being here. in the y-axis, what we can see is the CTNND2 expression. So how is that CTNND2 being turned on? So according to a control, it's set to 1. And what we see is, we see it in other genes too. Some guide RNAs might not turn off that expression, as is the case for like guide RNA 1, guide RNA 2. But something interesting that I want to throw your attention is guide RNA 3, guide RNA 6, and guide RNA 10 seems to be upregulating CTNND2 to a small extent. We really want to upregulate CTNND2 to therapeutic levels. So what I'm going to be focusing on next is trying to optimize the guide RNA 3, 6, and 10 to upregulate CTNND2 to therapeutic levels. But overall, we're really optimistic with the guide RNAs we have designed and the potential to upregulate CTNND2. The second thing is not only developing the therapy, but how do we get the therapy into the cells of interest? And for that, we use AAV. So what is AAV? AAV stands for adeno-associated viruses, and they were first discovered in 1965. After 1965, over 25 to 30 years, the research really focused on understanding the basic biologies of AAV. But in 1995, we had the first AAV human applications to treat cystic fibrosis. Then its clinical efficacy was tested further in 2008. And the two key deadlines that I want to really highlight here is 2012, with the first European Medicines Agency approval, followed by the FDA approval in 2017. So overall, AAV is the gold standard to deliver gene therapies in humans, and it's FDA approved, which is great. So one of the challenges with neurons, even though they're amazing, is that they're really hard to get things into them. And here we have an image of neurons under the microscope that we've been growing in Petri dishes. And it's a little hard to see, but what I have highlighted here is like that cell body. And then if you follow, there's some projections that are going off. And what I did to these neurons is I threw in some AAV that had this red fluorescent protein. So if the AAV got into the cells, it would fluoresce red. And that's what you're seeing on the right. And when I started doing these experiments, only, you can see here, only 2 cells out of like so many that are here were fluorescing red. So AAV was not getting really into the cells. And the cells are kind of hard to see because they're kind of unhealthy. They're not liking this virus going in them. So there's a little bit of cell death. So I spent months to try to optimize this condition, and I'm really proud to say that we have indeed optimized these conditions. So here you can see the structure of this neurons looks so much better. You see the cell body, and then you see these projections that are going off, and the neurons like to be, like the cells probably like to be together, so they're kind of grouped together. And you see a lot more cell dead, and you can appreciate the structure. So the cells are healthier. And also, we threw in that AAV, that fluoresces red, and what we see is we don't see 2 cells, we see a lot more cells. So the conclusion from this experiment is that after months of optimizations, we were able to optimize the AAV delivery into neurons. And that's something really excited because now we have a way to get this CRISPR-A therapy into the neurons, into the cells of interest. Finally, I want to think ahead, what are we going to be doing? And that is designing experiments to test this CRISPR-A therapy approach in patient-derived cell lines. So we have obtained 2 patient stem cells. And the wonderful thing about stem cells is that they have the potential to become any cell. And our strategy that we're thinking right now is to test this AAV therapy in brain organoids. So we've been in contact with the COSIC Lab in UC Santa Barbara, who are experts in neuroscience. And we hope for a collaboration in which we can test our AAV therapy, throw them in those brain organoids, and see how it changes the development of the brain organoids and seeing the rescue of CTNND2. The other aspect that we want to do is throw the AAV into neuronal cultures. So I'm looking at how to upregulate CTNND2 in the patient-derived cell lines. And after throwing our AAV into the brain organisms and the neuronal cell culture, we want to see upregulation of the CTNND2 gene, as well as measuring other cellular phenotypes, such as cell migration assays. overall, we're really excited with the progress we've made, and we're really looking forward to continue fine-tuning and developing this CRISPR-A therapy for CTNND2. Thank you so much for your attention and for your support.